So DNA is what we looked at the other day, and it's basically what you need to know if you want to understand genetics. So I'm going to try and do a really shortened version of what we did in class, just in case you need to refresh your memory. So remember that we have DNA, and it stands for deoxy, so not really requiring any oxygen at all, ribo. So we're looking at the proteins that form the sugar phosphate backbone. Nucleic, because it's in the nucleus, and acid. And it's found in the nucleus of the cell. So it's really protected in there. The membrane of the cell membrane is sort of small enough that the DNA can't get out, because we need to keep it protected. So nucleus of the cell. Okay. So if you recall the other day, just say we have the cell, whoops, right here, and inside the cell, this is all the cells in our body, we have the nucleus. Inside the nucleus we have what we commonly know as the double helix, so that's our DNA strand, yep. And then outside, in this little bit here, we have the cytoplasm. And that holds all the organelles in place so they don't get damaged. So the DNA is kept nice and safe inside the nucleus of the cell, so it can't get out. But if it can't get out, then how does it make the new cell? Because we need to transfer the genetic information from our original cell into the new cell. So if we quickly look at something called mitosis, we have our parent cell, and that parent cell divides to form two identical daughter cells. So the original DNA that's in the parent cell up here somehow needs to be shared so that both these daughter cells end up with the exact same information. So we need to look now at, okay, how we're going to get that DNA from the parent cell into the daughter cell. Okay, so that's fundamentally what we're looking at. So if you recall, we have our first stage, so we have stage 1. We'll quickly have that here. And we have our double helix. You've probably seen these pictures, all over the place. And then we have these little rungs in here. It's not the neatest on these iPads drawing these. Sorry about that. Okay, so this bit on the outside is the sugar backbone. And that is really, really strong, because we don't want that to be damaged, and we don't want that to break at all, because if it does, then the DNA is going to become damaged. These lines that I've drawn here are base pairs. Okay, and we have 4 of these bases, so we have 4 bases. And I'll write the names of those on the next section. So just note, again, we've got the sugar backbone, really, really strong. These lines here are our base pairs, and we have 4 base pairs. So if I was to draw that again really quickly, we have a DNA strand that I had before, the base pairs, sugar backbone, or sugar phosphate backbone, and our base pairs. So the letters of those base pairs: A, T, C, G. Okay, so we've got adenine, thymine, cytosine, guanine. Now these base pairs need to all add up. Now remember, I'm not expecting you to remember all these. So you just need to know that for successful replication to occur, we always need A joining up with T and C joining up with G. So if I was to quickly show you, so these base pairs will have an A and a T and a C and a G, a C and a G, oops, that's meant to be a G, a TA, so it doesn't have to be the same order all the time, as long as the T and the A joins together and the A and the C and the G. Alright, so before I mention the sugar phosphate backbone, these base pairs are joined together in the middle by bonds. Okay, and they're called hydrogen. So here we have a little bit of biochemistry in here. Okay, so those hydrogen bonds are really, really weak, and they're designed to break apart really, really easily. So when it divides, we can break that and we end up with two separate strands. So going into stage 2, we need to break apart that DNA strand. So it unravels and we end up with something that looks a little bit like this. Looks a little bit like a ladder. So we still have our base pairs along here. We still have our backbone. At this point, we still have our hydrogen bonds connected here. Hydrogen bonds. And we still have our base pairs along here, etc., etc. Alright, so if we go back to our little diagram that we had before, if we look at the DNA in the cell, okay, so it's in the nucleus of the cell, all this is happening, so we end up with the rung of the ladder. So I'm going to draw the cell again real quick over here. So we have our cell, I'm just going to draw it real big so you can see, and we have the nucleus in here. Now remember, at this stage, the DNA has unraveled and it looks like the rung of a ladder. So what happens is we get one strand breaks apart and it stays in the nucleus. That's one of your original strands. And the other strand that was part of that DNA comes out into the cytoplasm. So this is also an original strand. Now, during this process, we get a new nucleus that starts to form around there, the cell divides, and we end up with the two daughter cells. Okay, that's cell 1, and this is cell 2. Each of those cells contain an original strand of DNA that has the exact same information. So if we think back to our bases were, there'd be an A there, there'd be a T there, a C, a G, etc. G, whoops, C, T, A. Right? So these original strands become templates for the new DNA. Right? So now we'll go back to our strand and we're going to look at stage 3. Alright, so this original strand is from cell 1. This is the parent cell, and the other cell, the other original strand, is over here in a different cell. And basically what happens now, they become RNA. RNA, which is ribonucleic acid. Now ribo, remember, is from ribosomes. The ribosomes are proteins or make proteins, and those proteins actually is like an instruction manual Okay, so you go and you want to make something, you get something from, let's just say, IKEA, and it's not put together yet. But you get an instruction booklet that you read and it shows you how to put that piece of furniture together, hopefully. So RNA is kind of similar to a piece of IKEA furniture, the instruction manual. So it holds all the instructions via these proteins that helps to make the new strand of DNA. So we're going to go ahead and now the proteins that contain the instructions are actually going to make the new strand now, hopefully. Okay, so I wish we had some different colours here. Alright, so then those proteins come along and a new sugar backbone is formed. And with those instructions that are carried by those proteins, the original template strand is able to make a new strand of DNA. So now we've ended up with 2 copies of DNA, one for each cell. So this one is in daughter cell 1. That's daughter cell 1, and this is daughter cell 2 over here. So can you see the black strand is the original here, which carried the proteins as part of RNA to make a new strand, which is here. So if we think back to our original strand that was already carrying those base pairs from before, okay, so see how the black strand, the original strand, contains the bases. So we'll go C and a G, whatever. Alright, so now we just need to make sure that when it replicates, those base pairs actually match up to their correct partner. So we need to have a T form here, an A form here, a G form here, a C there, A, T, and a G, and a C, etc., etc. So can you see now that we've got 2 identical strands of DNA? Right, so if all goes well, hopefully if we get nice healthy cells, this all goes perfectly well and we get 2 perfectly good cells. Sometimes though, we might get what's called a mutation, and we're going to go into those a little bit later. But what a mutation is, when those base pairings are coming about, something happens in that makeup and we get something that shouldn't be there. So we know the rules with the base pairs. The A and the T always joins together, and the C and the G always joins together. But look what's happened. They aren't supposed to be together. So I was explaining before, the body has these awesome little, almost like proofreaders that come along. So when they're looking at the DNA strand, they come along and go, okay, that C is not supposed to be paired up with the T. So that's not right. We need to fix that, because if it doesn't, our genetic information isn't going to be passed across. So we can either end up with a bad mutation, which could cause, something like cancer or some kind of disease. It could be neutral, so it might not have any effect whatsoever. If it is a bad one, then hopefully the body will be able to fix it or destroy that cell, and then it won't divide again. So what the proofreaders do, they come along and go, yep, they aren't supposed to go together, so let's just get rid of that. That's not good at all. That's not good, people. That is not good. So we go A, back there again, or whatever it was, I can't remember. Oh, hang on. Oops. That's supposed to be an eraser. There we go. So we go back to having the T and then the A, and we go, oh, cool. That is exactly how it's supposed to be for a healthy cell. Okay, so remember in that first part of mitosis, I said that was the original cell. Yeah, one of the DNA strands, the original, stayed there, and this is what it was. And then the other cell, the other strand of DNA came out there. So that has now become into 2. Yep, each containing an original strand of DNA. And then once it's replicated, we end up, as you can see here, with that DNA strand raveling back up again, like this. Yeah. And let me go back to this. Can you see how that has happened? So every time you need a new skin cell or a new hair cell or a stomach cell or anything, liver, who knows, whatever, we need to get the same genetic information across. If we want our liver to be working, properly, or our kidneys or our stomach, skin, whatever, we need our good, healthy DNA to come across into the cells. We want to get an exact replication. So this ends up being stage 4. We've had one cell divided down the middle and become 2 cells, each containing an old strand and a new strand of DNA. Pretty cool, hey? And actually, you know what? Even cooler, the cell itself, so just say we have this original cell up here, you know that one cell can divide, it can carry out this particular division up to about 30 times, producing these extra cells, and then they'll eventually die. So each cell has a specific lifespan, and once it's divided enough, that cell's too old, it's not really needed anymore. We've got enough, hopefully, better cells or newer cells to continue on with that. So hopefully that's refreshed your memory from class. If it hasn't, please come and see me. I will go back over it in class. If you need to re-watch this, 10 more times, you can, or come and see me and I'll go through it with you.